Okay, so this is really the first lecture that I'm going to do on lab. I'm going to be very honest with you. This is the area that most of the AP1 students struggle in. That's histology. You generally at this point have heard of the bones, heard some of the muscles. You're familiar with some of the senses, but actually seeing tissue, most of you have not seen it. So it's going to be a little tough. Okay, so I'm going to go through this. I'm going to help you through this, and hopefully you will get there. All right, so first of all, the classification of the tissues. We classify tissues into 4 different groups: epithelial tissue, connective tissue, muscle tissue, nervous tissue. Epithelial tissue lines things, lines the inside, lines the outside. It lines organs, it lines ducts, it forms the skin, it's protective, it absorbs, it allows for filtration, it allows for secretion, it allows for excretion. So this is a very, very important tissue. Connective tissue, basically what I generally say is that it's a big group of tissues. If it's not epithelial, it's not muscle, it's not nervous, it fits into connective. So this is a protective, supportive type tissue. It's the most abundant tissue we have. It's going to underline a lot of these tissues, bind them together. So it's very, very, very good. But you're going to be surprised at the variety of those tissues that are in the group that's considered to be connective tissue. Because we have bone and we have blood. We have fluid connective tissue and we have hard bone. All right, muscle, that is that tissue that allows us to move. We have 3 different kinds. We'll talk about it. And the nervous tissue, that allows us to do these nerve impulses, allows us to make changes to maintain homeostasis. All right, let's start with epithelium. As I mentioned, this is a lining covering tissue. There is always what we call a free surface. We call this the apical surface or the apical edge, and there's always a free side that is exposed to the environment, doesn't touch anything. So it may be the inside of an organ, or it may be the skin or something like that, but there's always what we call this free surface, and that's what identifies an epithelial tissue versus these other tissues. These cells are very closely packed together. They are in sheets. They're connected by what we call these specialized junctions that we'll look at some of these, but it's a free edge. Once we get down below that layer, there is usually connective tissue. Okay, so the apical edge is the top of it. The bottom of that tissue is called the basement. This tissue has no blood supply of its own. It's getting the blood supply from the underlying connective tissue. So we call it avascular, but it does regenerate very well. Think about your skin. That is an epithelium. It regenerates extremely well, unlike something like a nerve that may never regenerate. All right, so when we classify epithelial tissue, it's 2 ways. And it's in the name. It's the number of layers and what they look like. Okay, so the first is the number of layers. If there's 1 layer of cells, it's called a simple layer. If there is more than 1, it is stratified. It may be 2 layers, it may be 25 layers, like 30 layers like skin. Okay, but you have a basement, you have a layer of cells, and you have the apical edge. That is the simple. Okay, so if we take a look at this, you can see. All right, so these are all simple layers. Now we're going to look into the shapes. We have a flat shape that's called squamous. We have, if you look at it going down on, and I'll show you the next slide, it looks kind of like a fried egg, but it's very flat, very thin. These kinds of cells are where we need to move things very quickly across certain areas, and we'll take a look at this and I'll explain this in a few minutes. But we want something very thin, very flat. So simple squamous epithelium. The next shape is cuboidal. It's a cube. The height of the cell is approximately the same as the width of the cell. So what you have to do is visualize that cell, look at the width, look at the height, and if it's approximately the same, then it is cuboidal. We generally do absorption and secretion with this. This is a smaller cell, so we can do a pretty good job with it. It's not made to do really a large amount of absorption, but it can do quite a bit. So this is a real nice general medium type cell. Simple cuboidal epithelium, and I'm going to talk about where these are found in a few minutes. And then if we really need to do a lot of work, we need a lot of more cell surface area. Okay, so we have to go to a bigger cell, and then we go to our columnar cells. So this is a column. The height of the cell is much larger than the width of the cell. So now we're going to look at these individually and take a look at these. So again, this is this simple squamous epithelium. Looking down on it, we see our lovely little fried egg, if you will. And these cells are stuck together and very tight. So we're looking at this simple squamous, and here we have an alveoli of the lung. This is where the gas exchange has to occur. And if you sit there and think about it, when we're talking about the respiratory system, we've got to move oxygen out into the atmosphere, into the blood vessels, and get it into our bodies, and we got to get the CO2 out. We have to have a very, very, very thin membrane, and that's it right there, that simple squamous epithelium. That is only the whole entire membrane plus the blood vessel is a half a micron thick. That's how small it is. And this simple squamous allows us to make these very small membranes that we can very easily move things across. Okay, let's look at another one. And here we have a couple of different epithelial cells in it. This is the glomerulus of the kidney. And you can see this is the glomerulus. This is where the filtration is occurring. So the blood's coming in here, it's being filtered here, and then it's moving into the tubules. So what you have is Bowman's capsule right around here. And if you look very carefully, you see these very thin cells. Those are the simple squamous epithelium. What you have to do when you're looking at epithelium, and this is the perfect example of it, is you look for your free edge. Here's a free edge. You go from the free edge down into the cellular tissue, or you're going from here down here, and you go down until you see a change. All right, so if I go down here, I'm going to come over here. Okay, so this is a slide of the glomerulus of the kidney, and you can see the glomerulus here. This you'll learn when we get to AP2 is the Bowman's capsule, and these are the renal tubules. And I like this slide because it shows you how to identify these tissues. So we just said that with an epithelial tissue you have to have a free edge. Well, this is a free edge. This is a free edge. This is a free edge. So you go in from your free edge and you look at the cell. So coming in from my free edge here, I have a single layer. It changes into something else over here. I don't know what it is, right? But coming in very flat, and then I change into something else. So we've learned flat cells, squamous, and we don't have another layer underneath this that's the same as this layer. So this is a simple squamous epithelium. When I get over here, let's go from this one. Here's our free edge. I come into this cell and I stop. I've got another free edge over here. But look at the shape of this cell. Here's the cell. It's a cube. And I don't have another layer of this underneath. So this is simple cubodial epithelium. And this is what makes up the glomerulus of the kidney. The actual filter in the glomerulus, this is this simple squamous epithelium, and you move into the tubules, and the tubules are simple cubodial epithelium. All right. Excuse me. So the simple squamous epithelium, we're going to see it in the glomerulus. We're going to see it in the air sacs, the alveoli of the lungs that we talked about. The lining of the heart, the endocardium, very, very, very smooth, so you have no clots. You'll see that in AP2. You'll dissect the heart, and you'll get to see that. Your blood vessels have to be very thin. You're trying to move white blood cells out into the tissue. The same thing for the lymphatic tissues. So this is a tissue that's used for diffusion, for osmosis, for filtration, where you have to move things very quickly, and you don't want any obstruction. The simple cuboidal epithelium, again, cube-like, usually in a single layer. There are a few places where you have stratified cuboidal epithelium, but we're not going to look at it here. So pay attention. Don't put that as an answer. It's in a single layer, large round nuclei, and the shape is a cube. And its function, as I said, is secretion and absorption. Not a large amount, but a medium amount, and it does fine. So you're going to see it in the kidney tubules. You're going to see it in some of the smaller glands, the surface of the ovary. But if we take a look at this, this is a little bit closer view of that tubule. You can see this is your free edge. There's the basement, the bottom of that cell, and then we go into another, but we have another free edge. So we basically have two cuboidal cells back to back with free edges on either side. So this is still a simple layer because this cell has a free edge and this cell has a free edge. But if we look at the shape of the cell, that's truly a cube. All right, so this is simple cuboidal epithelium. All right, so when we get to simple columnar, we're going to do secretion and absorption, but in larger amounts. This is found in the stomach, in the small intestine, the large intestine, where we're sitting there trying to get a lot of that gastric juice out and a lot of absorption done of our nutrients. Okay, some of the respiratory bronchioles, the endometrium has that. All right, but this is a column. So here we're looking at, this is the free edge. This is actually your intestinal tract. These are the villi, the little indentations you have in the gut. All right, and you can see the villi. This is the free edge right there. So you're going in from the free edge, and when you go in one cell layer thick, you change over to something else. So this is a simple layer. Well, now I have to decide what shape. Well, this is really narrow and really tall. So by definition, that is a columnar cell. Now we're going to look at another cell that's going to be very similar, and I want to point out something to you right now, and it'll make sense when we hit this other cell. I want you to notice how the nuclei are all kind of lined up. So if I drew an imaginary line through here, I would hit all the nuclei. Okay? That's very characteristic of simple columnar epithelium. One of the things that you do have to know for the test is this cell right here. This is called a goblet cell. Goblet cells secrete mucin, which becomes mucus, and it lines these tissues, and it basically Like, for example, in the gut, you have an alkaline mucus in the small intestine that the enzymes need that alkaline change, or you have a protective mechanism, the alkaline mucus in the stomach that protects the stomach from the stomach acid. So you may have different types of pH in the mucus, but it's a protective lining. Okay. All right. So that's our simple. We have one more simple, but it looks a little strange. We're going to move into stratified. That means there's more than one layer. The most famous stratified squamous is skin. We want stratified squamous where we need thickness. We need to protect against abrasion. These are areas where you get abraded, you get you're swallowing a lot of food, you're hitting a lot of things on your arm, that sort of thing. So you're getting a lot of abrasion, and you're going to have multiple layers, all right, of this squamous epithelium. So where you would find it, you'd see this is, again, the esophagus, where you're swallowing food, in the mouth, swallowing food, the vaginal area for sexual intercourse. You have to have that protection. Those will be what we call non-keratinized. They don't have keratin. But then we have the epidermis of the skin, which is keratinized. It has a keratin in it, protein. So here's this layer. And again, what you look at is, here's your free edge. You come in from your free edge. Well, there's a layer of cells. There's another layer of cells. There's another layer of cells. There's another layer of cells. So it's layer after layer after layer after layer after layer after layer until you finally get down to here. And then you change into a different type of tissue that we're going to talk about next when we get to connective tissue. But you can see that this is multiple layers of this tissue, and that is what skin is made of. This stratified layers of squamous epithelium. We're going to learn this. This is the stratum corneum. These layers have different names, but this is a very protective type tissue that we need to protect us against abrasion. Okay, then we have the oddball. Pseudostratified, usually ciliated, pseudostratified, ciliated columnar epithelium. Wow, that's a mouthful, okay? But it is a columnar epithelium. It is simple. It is a simple layer of columnar epithelium, but it looks like it's stratified. So we call it pseudostratified, pseudo meaning false. So simple columnar epithelium looks different, looks stratified, but isn't. Usually you will see it with cilia. It is found in the trachea, in the upper respiratory tract. And basically what you're using this for is, there's usually mucus, and we're going to do sweep and weep. We're trying to get all kinds of nasty things out of our lungs. So let's look at this. This looks different. We're coming in from the free edge. We're going through this layer, and here's your change right here. This is your basement. But, geez, I don't have a free edge here. I got all these nuclei all over the place. And there's my little cilia. But if you remember what I said about the simple columnar epithelium, what I said was you could draw an imaginary line and you'd hit all the nuclei. Well, in this one, you hit this line, and you hit this line, and you hit this line. So it looks like you have a stratified tissue, but you don't. It's just that the nuclei are at different heights. Okay, and this is very characteristic of that. All right, so how to tell simple versus stratified columnar? I've got that picture for you to make it easier. I got the two together. So there's our simple columnar, all those lovely little nuclei in a row, versus the pseudostratified. There you go. Okay. It's not real stratified, it's pseudostratified, it's one layer. Okay, so don't get confused. And then our last is transitional epi. This is a stratified squamous. It's transitional because it transitions shapes. It changes shapes. The cells are round when the organ is empty. They flatten out when it begins to distend. Think of the bladder, think of the ureters, think of the urethra. When the bladder is full, it is flat. When it is empty, these cells are more round. They look more cuboidal. Okay? So you can look at it here's this is a pretty smooth edge here, and this is all kind of bumpy here. And that's how you tell the difference. Okay? All right, so required for the exam, you need to know these tissues. For your lab practical, this is again for the lab. So you need to be able to recognize these tissues. You will have a picture of them. You will be required to identify them. You will be required to identify a goblet cell. Anything on this list that says you got to know this, you got to know these. So you have to be able to identify them. And all these tissues could be on the test. You have to be able to recognize them. You have to know where they're found, and you have to know their function. But I have a worksheet for you. Okay, so that worksheet will help you through this. All right, and that's it for epithelial tissues. I'll see you next in connective.